Okay, I'm assuming you're all energized and ready to go for the last stretch run. I'm not sure. Okay, I will have to close the lab tomorrow afternoon after lunch to get lab exam set up. Everybody's got the study guide for the lab exam. So if you have any last minute questions, tomorrow would be your last chance to ask them. Okay? Okay, so we're in the stretch run on the immune system, excuse me, and we're talking about the third line of defense. Remember that it's the third line of defense that's characterized by being acquired. This is not something you're born with. This is something you develop in response to exposure to pathogens. It is also characterized by being adaptive in that it changes and evolves during the process of encountering and dealing with the antigen. And it also is associated with memory. So once you have been exposed to an antigen and hopefully survived, that you will then be better equipped to deal with it in future encounters. And that's the basis of immunization. Pretty complex interaction taking place between a variety of immune cells. And one of the things that we're going to see is that there's a lot of chemical signaling that is involved. In order to understand the complexity of that chemical signaling, these are some of the topics that we're going to have to deal with first in the next couple of class periods to make sure you kind of understand what's going on and how these important immune cells are functioning. We're going to be looking at the major players being macrophages and their cousins, if you like, the dendritic cells, B lymphocytes, and T lymphocytes. So just the interaction between those cells is going to be pretty complicated. There are various cell surface molecules we'll talk about, including MHC molecules. We'll tell you what those are in a second. B cell receptors and T cell receptors. Those are found respectively on B and T cells. And then there are some other molecules we'll briefly mention called CD receptors. Additionally, we're going to have to talk a little bit about some of the common cytokines that are involved in the chemical signaling between these immune cells. We also then will have to know what antigens are, because antigens are what stimulate this whole process. And we'll also have to know what antibodies are, because antibodies are one of the cell's responses, or one of the body's responses, to immunological challenge. If you're following along in notes, the first section of the line 3 notes is kind of an overview. And that goes along with the first 3 or 4 slides, which is intended to kind of set the stage for you. And these are notes and slides that I would encourage you to go back to periodically and reread to kind of reset the big picture, because as we dig into more details, sometimes it's easy to lose the forest for all the trees. And I'm very interested in you understanding the forest and that big picture and how things fit together. So this slide, for example, this slide shows you kind of overall, 3 major events that occur during the line 3 immune response. And one of them is going to be generation of the major types of cells, the B lymphocytes and the T lymphocytes, their education, how we get rid of cells that we don't want to have that might react to self, how cells that survive might then respond to antigen. So basically generation of the immune players up here. Down here, and this is an antigen-independent response, okay? You don't need the antigen for this to happen. This is happening all the time. Down here is an antigen-dependent response, and this basically talks about how these immune cells that were generated here become activated in response to a threat. And one of the things that we're going to see is that antigen-presenting cells, this is a dendritic cell up here, play a really important part in most of the activation of these cells. So we've got activation of B lymphocytes, we've got activation of T lymphocytes. Down here, we've got kind of a split. And that is indicative of the fact that the immune system, line 3, is generally considered to be consisting of two functional parts. And we call those the humoral immunity and the cell-mediated immunity. Humoral immunity is primarily the province of B cells, although there is T helper cell involvement in this as well. We'll talk about that later. But the outcome basically is, as a result of the activation of these B cells, by the appropriate signals, they start producing antibodies. Now, we haven't really gotten into what antibodies are and how they work, with one exception. And that is, we said, gee, if you have an antibody, that could stimulate that second line of defense mechanism, the complement system. So that's one of the ways in which antibodies are beneficial. But there are other ways as well. Simultaneously, we've got this cell-mediated immunity response going on. And that involves T cells. These are involved in killing your cells that have become damaged. Typically either cancerous or virus infected. These two arms of the immune system work functionally together to eliminate pretty much any threat that you might be exposed to. Now, in the text of the notes, if you have those printed out, there is some little bit more detailed explanation about what's going on in here. But again, it's always a good idea to come back to this so that when we start talking about more detailed concepts, you can say, okay, yeah, that's part up here. Okay, that belongs to this part of kind of the big picture. And then when we talk about something else, oh, that's down here. That belongs to this particular part of the big picture. So I really like to kind of take a couple steps forward and then occasionally go backwards and kind of relate it to the bigger picture. And then these slides are just kind of the same thing, just another way to represent the varieties of interactions that take place. So these are considered to be summaries. So let's dig in first to this. Let's start looking at cell surface molecules. One of the things that I hope you come to appreciate in going through this immune response information is that I think the common perception of a cell surface is that it's just this kind of lipid bilayer, and it's kind of a naked lipid bilayer. There might be some occasional molecules sticking out on the surface that are receptors. But I don't think anybody who hasn't studied immunology can ever appreciate the complexity of receptors that are associated with the surface of a cell. Those receptors, in many respects, give that cell identity. And one of the things that's important for is it allows the recognition between self and non-self. This belongs, this doesn't belong. So that's one of the major roles that some receptors play, but it's not the only role. Another role is that if receptors are bound by some type of a ligand, typically an antigen, that is kind of an activation signal. And the activated cell can start producing various lymphokines. These are chemical signaling molecules. The other thing that activation of these cells can result in is changes in cellular development. For example, when a B lymphocyte is first produced in the bone marrow. It has to go through an education process first, and we'll talk about what that education process involves. But just because you're educated doesn't mean you can really do anything yet or are worth anything. Typically, you don't really become a player until you've been activated. We call those educated cells that really haven't done anything yet naive cells. Okay? That's kind of like you when you finish college and go to medical school. Or maybe even when you finish medical school before you were starting your residency. You know stuff, but you've never really done it. Okay? But as a result of activation, which involves something binding to a receptor, signals are transmitted throughout the cell, which result in cell differentiation. And that naive cell, that naive B cell, now differentiates further into an active antibody-secreting cell called a plasma cell. So this can now secrete antibody. The educated naive cell knew how to do it, but it hasn't done it yet. It hasn't received the proper signal. The amount of antibody that these cells can secrete is incredibly impressive. Thousands of molecules per second. They're just cranking this stuff out. And in many respects, they just kind of burn themselves out. Well, one of the developmental changes that occurs within these cells is that there's a massive increase in the amount of membrane-bound ribosomes and rough endoplasmic reticulum. Those are all associated with secretory proteins. Okay? So these differentiated cells have a different number and kind of organelles than the naive cell does. They have much more rough ER. What triggered that developmental change? Binding of an antigen, or maybe some kind of a signal received from another cell, some kind of a chemical signaling molecule from another immune cell. So these receptors are really incredibly important. MHC receptors were first discovered in this context of identity and self versus non-self. MHC receptors are also sometimes referred to as HLA receptors. HLA stands for human leukocyte associated. They were first discovered in blood cells. The context that these were really most important in initially when they were first discovered was they explained something very important. And that is why sometimes transplantations of organs and tissues took, and why other times they didn't. There was a rejection. As it turns out, everybody's cell surfaces are loaded with these MHC molecules. All cells in your body have MHC1s with one exception. Red blood cells don't have these. Red blood cells are unique in another way. Remember, they're non-nucleated. Red blood cells are kind of atypical to our cells, but they don't have MHC molecules. All cells in the body, except red blood cells, have MHC1s on their surface. So this gives identity. Okay? And if we did an MHC profile of Jacob's cells and one of Katie's cells, they would have different profiles, and especially if they were not related in some way. Because receptors are proteins, and proteins are determined by genes. Okay. So what they found out in tissue transplantation was donors and recipients that were more similar in their profile of MHC1 had a greater chance of success of transplantation taking. If there was more dissimilarity in their profile, then there was less of a chance. Well, the reason is because great dissimilarity in these receptors or cell signatures triggered the immune response. And as it turns out, it was a T cell response. And those cells were killing the transplanted tissues. MHC2s weren't discovered until much later. And they're only found on certain cells, those that are capable of presenting antigen. APC stands for antigen presenting cell. Antigen presenting cells include macrophages or dendritic cells, kind of the same. B lymphocytes can also be antigen presenting cells. So these are really key players in the immune response. And they're kind of identified or marked by having these special groups of receptors called the MHC2s. So, class 1 MHC found on every nucleated cell. Class 2 are found only on B cells or other types of antigen-presenting cells. The structure of the receptors are a little bit different. Both of them have a little groove in which particular antigens can bind to these particular receptors. So let's briefly take a look at class 1s first, MHC1s, and see what they are and what they're doing. A real key to the immune response is antigen presentation. Basically showing an antigen to other cells so they can see what it looks like. There are two major routes. Intracellular and extracellular. Class 1 MHCs are involved in the intracellular antigen presentation. Class 2 MHCs are involved in the extracellular antigen presentation. So, all cells in your body, except red blood cells, have these class 1 MHCs. Where are those receptors located? Well, actually, those receptors are located on the membranes of the endoplasmic reticulum. Cells are constantly producing proteins, right? Their genes are good, their genes are active. And those proteins are constantly being broken down or catabolized. Remember the concept of protein turnover? Proteins are made, they're degraded, they're made, they're degraded. So during the process of protein turnover or catabolism, some of those fragments of the proteins that the cell normally produces, can enter the membranes of the ER, associate with these class 1 MHC molecules, vesicles pinch off. They can go to the Golgi first, but eventually those vesicles fuse with the cytoplasmic membrane, and now these MHC protein fragments are displayed on the surface of the cell associated with this MHC1 receptor. Remember here we've got the receptor in the little groove. So here's where the piece of protein would be displayed. Right here. So little bits and pieces of all the proteins you normally make are displayed on the surface of your cells. Think how crowded the surface of the cell must be, because this is only one kind of cell surface molecule. With the cells loaded up with these MHC receptors, displaying pieces of normal polypeptides that the cell is constantly making. This then gives that cell surface identity, a signature. And because Jacob is producing slightly different proteins than Casey, then his MHCs will look different than hers. Tissue typing can be done to identify an individual's MHC profile. And that's what they do when they try to determine suitability for organ transplantation. Is they tissue type certain key MHC antigens and look for levels of similarity and dissimilarity. And if they're too different, then you're not a good match. Again, that's why families are oftentimes the best donors, because they share common genes. If they share common genes, they produce common proteins, and therefore their cell surface should look similar. So this is a way to give identity. Okay? Now, one other thing. If a cell starts producing some abnormal proteins, because it's been infected by a virus, or it's become cancerous, those can become part of your cell surface. Now your immune system can look at that cell and go, doesn't look quite right. This is not what I would normally expect to see. And therefore, an immune response might be mounted against that altered cell. Typically, the immune response that would be mounted would be a T cell response, where one of these T cells would kill that infected cell. You don't want it to divide, because it'll either spread on the virus or it'll spread on the cancer. Okay, class 2s. A little bit different structure. They are found in vesicles found in the membrane of antigen-presenting cells. So this might be a dendritic cell, this might be a macrophage, or it could be a B lymphocyte. So they ingest the foreign antigen. It'll become enclosed in a vacuole that fuses with the lysosome. Remember that combination is called the phagol lysosome. And then the foreign antigen is kind of pulled apart. And there are now pieces of that inside that phagol lysosome. That then can fuse with this vesicle containing the MHC proteins. And now these pieces of foreign molecule become associated with those MHC2s. This vesicle then fuses with the membrane, and now on these kind of cells, sticking on their surface are the class 2 MHCs that are bearing foreign pieces of molecule. Okay? Class number 1 is typically self, unless the cell's been altered. Class 2 are showing foreign. Now, this is going to be really important. When we talk about T cells, T cells are involved in a process called cell-mediated immunity. And in cell-mediated immunity, immune cells are killing host cells. They're killing bad host cells, cells that have become altered. Most of the killing involves these cells called T killer cells. Those particular T cells are also called T cytotoxic cells. Cyto meaning cell, toxic meaning killing. But there are other types of T cells as well. There's some cells called T helper cells. And they're going to be involved in helping B cells produce antibodies. Haven't gotten there yet. And then finally, there are T suppressor cells. And they're going to be responsible for shutting down the immune response. Once it's worked, you want the response to stop. T cells are said to be MHC restricted. What restriction means is that they can only see or detect the antigen if it's associated with an MHC receptor. So think about the implications of this for a second. If there is a virus particle floating around in your blood, does it have foreign molecules associated with it? Absolutely. And will it stimulate an immune response? Yeah. But that free-floating virus will not produce a T cell response because they can't see those antigens, because they're not complexed with that MHC molecule. Now, if the cell takes them up and degrades those antigens and sticks it on the surface of an MHC, now the T cells can see it and respond to it. T helper and T suppressors are MHC2 restricted. So they can only see antigens in the context of MHC2s. Which cells have MHC2s? Macrophages, B lymphocytes. T killer cells are MHC1 restricted. So anytime a foreign antigen gets stuck on the surface of a cell with a class 1 MHC, a T killer cell can see to that and respond to it. So this allows different types of T lymphocytes to respond to different types of cells depending upon the type of receptor that the antigen is associated with. Now, that concept of class 1 and class 2 MHC restriction is pretty important. An additional type of receptor that is associated with immune cells are called the CD receptors. And about all I want you to know right now is that different types of immune cells have different types of CD receptors. And probably the two CD receptors that I'll refer to at all will be CD4 and CD8. Among others, T killer cells have CD8 receptors on their surface. It's not the only one, but they definitely have this one. T helper cells have CD4s. The reason that I tell you that is that sometimes T helper cells are actually referred to as CD4s, and T killer cells are referred to as CD8s. One of the things that you're very, very interested in if you're a physician treating an HIV patient is their CD4 count, T helper cell count. Because HIV specifically infects T helper cells and kills them. And so one of the things you see is a real decline in the T helper cell population. Well, that's pretty damaging, because these T helper cells help B cells produce antibody. So HIV knocks out antibody production. And that's why HIV sufferers are typically suffering from diseases that the immune system normally will deal with. But they've got a depressed immune system because those T helper cells, or CD4s, have been killed by the virus that's infecting them. Okay, so you can see here are some of the types of cells we're going to be dealing with. A B cell, these are some of the CD receptors that are associated with it, CD40 particularly. There are two types of T helper cells we'll see, T helper 1 and T helper 2. They both have CD4s, but then they differ in some other CD receptors. Cytotoxic T cells have CD8s, among others. The regulatory or suppressor cells have CD4s, among others. So don't try to memorize all this information, but just realize that the type of CD receptor a cell has puts it in a particular population of immune cells. And probably you should remember this CD4, CD8. Okay. Other types of cell surface receptors. The BCR. The B cell receptor. The cell surface of B cells is loaded up with these B cell receptors. There's 50,000 or more copies, identical copies of a B cell receptor on every B cell in your body. Remember, receptors is kind of adding up, isn't it? Okay, because these will also have CD1 or MHC1s loaded up with all the normal pieces of protein that the cell produces. And they've got all these B cell receptors. They've got some CD receptors. It's getting pretty crowded out there. What do these B cell receptors look for and bind to? A specific antigen. That specific piece of antigen is called an epitope. File that away for later on when we talk about antigens. Every immune cell in your body that's a B cell, every different types of B cell is a different B cell receptor. It has one kind of receptor, and that will react with one type of antigen or epitope. The process of making this receptor is what I've referred to as education. So the education process involves building a unique receptor. And once you have become educated, if your B cell is bone marrow, and if your cell type and receptor type has been deemed not to be one that reacts to self. Well, if it does, you better eliminate it. So we'll talk a little bit about clonal deletion in a second. But if the cell survives, then it migrates out into the lymphatic system and takes up residence in one of those secondary lymphoid tissues, like the lymph nodes. It sits there. And it waits. And it waits. Until someday it may be presented a foreign antigen. One that matches its receptor. That antigen is going to be shown to it by an antigen-presenting cell. So the antigen-presenting cell is going to engulf that antigen, stuck it together with the class II MHC, and put it out on its cell surface. Now that T helper cell, or that B cell, which is MHC2 restricted, can see that antigen. And if the receptor matches it, a binding occurs, and all kinds of weird and wonderful things start happening to that B cell. The most important of which is it starts producing antibody. And the antibody is protective. This idea that a specific antigen binds to a specific receptor is really, really important. So the specificity between a particular B cell and a particular antigen is really critical to understand. Now that introduces some interesting dilemmas, if you think about it. There are a whole lot of different antigens out there in the world. So what does that imply? That implies that there has to be a different B cell for each and one of those different types of antigens. That means there's got to be a whole lot of different types of B cells. And since an antigen is specific for a particular receptor, each one of those B cells has to have a unique receptor. So there's got to be a whole lot of different kind of receptors. How do all those arise? Where did all those different receptors come from? We'll get there in just a second. Okay, here is a diagram showing you the basic architecture of a B cell receptor. And there are some molecules that are anchored into the membrane, but here's kind of the guts of the receptor for B cells. The ones in green are called the heavy chains. The ones in yellow are called the light chains. So there are heavy and light chains for the receptors. Actually, two of each. Each one of these polypeptides, both heavy and light, are coded for by a variety of genes. There are actually 4 genes that are involved in the coding of a heavy portion of a receptor, and they're designated V, D, J, and C. V stands for variable. D stands for diversity, J stands for joining, and C stands for constant. The light chain is also encoded by genes, but only the V, J, and C genes. So there's no diversity genetic component of these light chains. Think of these as little genetic modules. So there are these V modules, D modules, J, C. And there's more than one of each type. There's more than one type of V. There's more than one type of D or J or C. But if you stick 4 of those together to make a message and then transcribe that message, you will make a heavy polypeptide chain. You'll make that part of the receptor. There are other modules that make up the light chain, the V, J, and C. And again, if you stick a V, J, and a C together and transcribe them, then you make a light chain. So this is what is happening in the bone marrow during that process of education, as the cells are building receptors. Let's come back to T cell receptors first. Let's continue on. Okay, so here's our dilemma. What we have is we have all kinds of different foreign molecules that your body should be able to respond to. Foreign antigens. How many foreign antigens are out there in the world? Well, probably billions. If you want to respond to that antigen by producing an antibody, then you've got to have a unique B cell that will match the structure of that antigen. The B cell receptor will match the antigen. Where does that matching take place? Up in this region right here of the B cell receptor. Actually, the B cell receptor has 2 antigen binding sites. It's got one here, and it's got one here. The number of molecules of antigen that a receptor can bind is called the valence. So this has got a valence of 2. So every different kind of B cell's got a different receptor. So if there are billions of different types of receptors, there would have to be billions of different types of B cells. Because what's the alternative? The alternative is that there's a single type of B cell, but on its surface, every B cell has a billion different kinds of receptors. There's not room for that. Okay? So how many genes is it going to take to build a different receptor for billions of different B cells? You'd think a lot, right? Well, how many genes are there in the human genome? Well, we keep revising it downward. Now the estimate is there's somewhere between 20 and 25,000 genes, functional genes in the human genome. That's not nearly enough genes to build all of these different kind of receptors for all the different types of antigens. As a matter of fact, when you dig into the genetics of it, there are only, a couple of hundreds of genes that are actually involved in making B cell receptors. So with that limited amount of genetic information, how can you generate all of this diversity of B cells to respond to all the diversity of antigens that are out there in the world? Well, the understanding of that was really a mind-blowing experience for most geneticists. Because for a long time we had this misconception about the genome of human cells, particularly of those stem cells that give rise to lymphocytes. We tend to think of genomes as being pretty stable. And one of the things that we see now happens during education is a phenomenon takes place that we call gene shuffling. Two keys to gene shuffling are the existence of RSS sites RSS stands for recombination signal sequences. And RAG enzymes. RAG enzymes are recombinant activating gene proteins. What these enzymes do when they recognize the proper signals, the proper sequences within a gene, is that basically they dice and splice genes. They cut out gene segments and stick them together. What kind of gene segments are they cutting out and sticking together? The V's, the D's, the J's, the C's. And if they cut out one of each of these and stick them together, what do they have? They've got a heavy polypeptide chain. They've got half of that B cell receptor, basically. Or if they dice and splice a B, a J, and a C, then they've got a light polypeptide chain. So, here's what the genome might look like. Located within your genes, here are some of the heavy chain gene loci. V stands, there are 65 of these different V genes, or little building blocks, if you like, for making receptors. There are 27 different diversity genes. There are 6 different joining genes. And during the process of education, that RAG enzyme recognizes the RSS sites that kind of separate these gene blocks and says, okay, I'm going to chop out one of these D's, one of these J's, and one of these V's and stick them together. So there's actually a genetic rearrangement of the DNA in these cells. And then, if it's stuck together with a constant gene, then I've got a heavy polypeptide chain. If it's making light polypeptide chains, it'll cut out one of these V's and one of these J's and stick it with another C and make a light chain. Now, this gives you an idea of where some of the diversity comes in. Light chains, as it turns out, are of 2 types. There are kappa chains and lambda chains. So this shows you lambda light chains, kappa light chains, and then heavy chains. How many of the different genetic elements do they have? Well, if you look at the lambda light chains, there are 30 different V regions. There are no D regions. There are 4 J regions. So these are the number of combinations you can get of V and J. Light chains have this number. So these are the number of combinations. When you're building a receptor, you can use either a light chain kappa or a light chain lambda. It's completely random. So that gives you some new combinations. Heavy chains, there are 100 V regions, 27 Ds, 6 Js. So with all these different combinations of V, D, J, and C, this is the number of different combinations you can get. 5 times 10 to the 6th. That's not a billion. So this is only where part of the genetic diversity is coming from in making those receptors. We'll look at next time some other factors that play into generating receptor diversity. But this gene shuffling, it's kind of like having a deck of cards. How many different hands can you deal in poker of 5 cards with a 52-card deck? Depends on how you shuffle it and what you deal out. That's exactly what's happening within the genome of these cells, is that we're shuffling the genes and dealing out cards which are polypeptide chains that are part of receptors. It's really a very cool process. Okay? So, we'll continue.
Can you tell us what all this is
Wednesday, I think the only thing that is due is your one ID factor, is that correct?
Yeah, what about our grouping list?
That's through the last day class.
Okay, and then
And for ID factor, all I need to know is your name, your number, and what computer it's on.
Yeah.
Just the one assigned one. The optional ones are due to the last day class.
Okay.
When you say Monday class, you mean your final day or the last day of lecture?
Last day of lecture, goes to the Friday. Yeah. Basically, everything's due then except your final exam.
Dr. Browder,
Yeah.
I'm applying for optometry school.
Mhm.
I was wondering if you could possibly write me a recommendation.
Sure. Would you send me the most recent copy of your resume?
Okay.
And do you have to do any kind of personal statement or anything for optometry school?
I don't think so.
Okay, okay. So if there's anything you think I should be aware of that wouldn't be on your,
Resume?
yeah,
Okay.
then let me know.
Okay. Thank you very much.
Sure.
The ID factor project, right?
Just the one required one. The optional ones aren't due till last day class.